Cell division meiosis I will focus on the fundamentals of meiosis, particularly the movement of chromosomes through two cell divisions, resulting in gametes that have half the number of chromosomes as the original cell. Note that meiosis produces gametes that are genetically different from one another. The details of the genetic variety that results from meiosis will be the subject of meiosis II. The essential ideas are: chromosomes carry genes in a linear sequence that is shared by members of a species, and secondly, alleles segregate during meiosis, allowing new combinations to be formed by the fusion of gametes. Meiosis is a highly specialized cell division that occurs only in the sex organs, the testes and the ovaries of mammals, unlike mitosis, which can occur almost anywhere in the body for the purposes of embryonic growth, growth toward adulthood, and tissue repair. Meiosis only occurs in sex organs. As well, meiosis produces gametes, sperm and egg, each with half the number of chromosomes of other cells in the body. Meiosis is a reduction division of a diploid nucleus to form haploid nuclei. The condition of having half the number of chromosomes, one set of 23, rather than two sets of 23, is known as the haploid condition. Also keep in mind that gametes are genetically different from one another, producing great genetic variety within a species. How meiosis produces genetic variety will be the subject of Meiosis Part II. Here are the IB syllabus statements that kick off this movie. State that meiosis is a reduction division of a diploid nucleus to form 4 haploid nuclei. State that the halving of chromosome number allows a sexual life cycle with fusion of gametes. Meiosis reduces the chromosome number to form haploid cells known as gametes, and then gametes fuse during fertilization to restore the diploid chromosome number. Meiosis is quite different from mitosis. Meiosis occurs in sexual organs only, whereas mitosis can occur nearly everywhere in the body. Meiosis has 2 cell divisions. Mitosis has only 1. Meiosis reduces the chromosome number. Mitosis does not do this. And meiosis produces genetic variety, while mitosis produces cells that are genetically identical. The genetic variety among the gametes that form from meiosis comes from crossover and independent assortment. And while Meiosis Part II will focus on these processes, be alert, because this movie will introduce important foundation for understanding crossover and independent assortment. If the language here feels quite unfamiliar to you, pay attention near the end of the movie to gain control of it. The explanation of how sexual reproduction, how meiosis promotes variation in a species, will be the focus of Meiosis Part II. All of the cells of your body have 46 chromosomes, 23 pairs, with the exception of sperm or eggs. The pairs come from the fact that you have inherited 23 chromosomes from your father and 23 chromosomes from your mother. Think of inheriting 1 set of chromosomes from your mother and 1 set from your father. 23 chromosomes would be 1 set of chromosomes, while 46 would be 2 sets. Sperm cells and egg cells carry 1 set, 23. This condition is known as haploid. Once fertilization occurs between 2 haploid gametes, the diploid, or 2-set condition, is restored. Meiosis reduces the chromosome number so that when fertilization occurs, the diploid number is restored. If meiosis did not reduce the chromosome number, then fertilization would result in a doubling of the chromosome number, a doubling with each generation. Doubling the chromosome number cannot happen if a species is to maintain its genetic integrity. Here is a slide to check your understanding. Turn the movie off now or follow along with me. Event A is meiosis, producing haploid cells 1 and 2, which are egg and sperm respectively. Haploid cells, sperm and egg, fuse in an event known as fertilization. Fertilization restores the diploid condition in a cell known as the zygote. Mitosis is the event of development. All of the cells of your adult body are diploid, with the exception of sperm cells or egg cells. Gametes, sperm and egg, have half the number of chromosomes, or one set of chromosomes, relative to other body cells. Why is it critical that gametes carry half the number of chromosomes of other cells? If meiosis did not reduce the chromosome number, fertilization would result in a doubling of chromosome number with each generation. The halving of chromosome number allows a sexual life cycle with fusion of gametes. This graph provides an important way of viewing meiosis, the amount of DNA per cell over time of two cell divisions. Notice that DNA replication also occurs before meiosis, just like mitosis, but notice that there are two divisions, resulting in cells that have half the quantity of DNA per cell as compared to the original germinal cell at G1. In fact, the gametes here have half the number of chromosomes as compared to the germinal cell. Gametes are haploid with 23 chromosomes in humans, one set, no homologous pairs. Lastly, I've put this small line here to indicate that meiosis is not a cycle of cell division like mitosis. Gametes have one of two fates. One, they fertilize another gamete, or two, they do not. Once a gamete is formed, there are no more cell divisions of that cell. Here is a nice comparison of mitosis and meiosis. DNA replication occurs in both processes before the cell division. In this case, the original cell in each example is diploid with two sets of chromosomes, 2n. But meiosis has two divisions, as can be seen here, resulting in 4 daughter cells. Each daughter cell is haploid with one set, 1n, of chromosomes. But remember, gametes are genetically different from one another. Mitosis results in 2 daughter cells, both, in this case, diploid, 2n, and both genetically identical. You've seen this image before. It shows DNA replication, where the chromosome becomes a 2-chromatid chromosome prior to cell division. Remember that chromosomes have genes, which are sequences of nucleotides that code for the building of protein. Protein is the expression of a gene, and we inherit the codes for genes in the sperm and egg from which we were conceived. Keep in mind that DNA replication occurs prior to mitosis and meiosis. As a cell in the ovary or testes enters meiosis, every chromosome is a 2-chromatid chromosome, seen here. This is just like mitosis. We can see many 2-chromatid chromosomes here with a light microscope following condensation during prophase of the first meiosis division. This diagram is the X-shaped structure seen immediately after replication. The sister chromatids are genetically identical, but once crossover occurs, the sister chromatids are no longer identical. Now keep your eyes open on this. I will cover crossover well in Meiosis Part II. Now here are 3 IB syllabus statements, very important ones that focus on homologous chromosomes. Homologous chromosomes carry the same sequence of genes, but do not necessarily carry the same alleles of those genes. Diploid nuclei have pairs of homologous chromosomes. Haploid nuclei have one chromosome of each pair. Remember that chromosomes exist as pairs in diploid cells, like all the cells in this man's body, the man who provided the chromosomes seen on this slide. Pairs exist because we inherit one of each pair from our father in the sperm and one of each pair from our mother in the egg. Remember that fertilization restores the diploid condition. Homologous pairs, as seen here, carry genes for the same characteristics, and we inherit a code for the gene from our father in the sperm and another code for the same gene from our mother in the egg. The codes for the gene are located at the exact same location known as the locus on the chromosomes. Alternative codes for the same gene are known as alleles. We inherit an allele for every gene from our father and an allele for every gene from our mother. But the 2 alleles for a gene that we inherit from our mother and father are not necessarily identical. They could be, but they're not necessarily identical. Alleles are alternative codes for the same gene that share the same locus, in other words, the same location on the chromosome. Homologous chromosomes carry genes for the same characteristics. Homologous chromosomes pair up during meiosis. They pair up literally, physically, during meiosis. The homologs come together and intertwine during meiosis. Homologous pairs are the same size, as you can see here. The centromere is in the same position, as you can see, and the staining pattern is the same for each chromosome of the pair. In the slides leading up to this one, I've focused on homologous pairs. Maybe now you will better understand my long lead-in to meiosis. Here are the relevant IB syllabus statements. Outline the stages of meiosis, clearly stating the events of each stage. Mention should be made of homologous pairs of chromosomes, 2 divisions, diploid and haploid cells, and the outcome of meiosis. As well, can you describe the behavior of chromosomes in the phases of meiosis, including crossing over, condensation, and the random orientation of homologous pairs at meiosis I metaphase. The processes of crossing over and the random orientation of homologous pairs will be covered well in Meiosis Part II. Be patient, take it one slide at a time. But here we go toward an understanding of the stages of meiosis and the behavior of chromosomes in the phases of meiosis. In this image, the first division of meiosis is outlined. In prophase I, the chromosomes condense. Then the 2 chromatid homologous chromosomes pair up. Crossing over happens here in prophase I, and because of different alleles on homologous pairs, sister chromatids will no longer be identical following crossing over. More on crossing over in Meiosis Part II. In metaphase I, the homologous pairs align together and move together to the metaphase plate. Spindle fibers, one on each chromosome, move the pairs to the cell equator. Notice only one spindle microtubule is attached to each chromosome. Anaphase I results in homologous pairs being pulled to opposite sides of the cell. One spindle microtubule per chromosome. Notice that the sister chromatids are not separated here. In telophase I, we have the end result of 2 cells, each with half the number of chromosomes of the original germinal cell. Count the chromosomes to understand this well. 1, 2, 3, 1, 2, 3 in each cell, whereas this cell had 6. This cell was diploid, but following meiosis I, there are only 3 in each cell. These cells are haploid, but the sister chromatids have not yet separated. This is the role of the second cell division, meiosis II. In meiosis II, the second cell division, starting with 2 cells, the chromosomes condense in prophase and attach to spindle fibers. In metaphase II, the spindle microtubules move the chromosomes to the cell equator. Notice that there are 2 microtubules attached to each 2 chromatid chromosome, one on each side. With this, you can anticipate anaphase II. In anaphase II, the sister chromatids are separated. Keep in mind that they are no longer genetically identical at this stage because of crossing over in prophase I. And then in telophase II, the nuclei become distinct as the chromosomes decondense and new nuclear membranes form. There are now 4 daughter cells, each with half the number of chromosomes of the original cell. Each cell is haploid. So let's review. Prophase I, chromosomes condense, homologous pairs come together, intertwining in a structure known as a tetrad. Regions of overlap, seen here as chiasmata, are locations where crossover occurs. Metaphase I has the homologous pairs aligned together, being moved in pairs to the equator of the cell. 1 spindle microtubule per 2 chromatid chromosome. In anaphase I, homologs are separated, but sister chromatids are not. In telophase I, new nuclei form. We have 2 haploid cells produced. Now, pay close attention. I will not display another set of slides showing the second cell division of meiosis. The second division of meiosis follows the same pattern as mitosis, where sister chromatids are separated by spindle microtubules attached to each side of the two-chromatid chromosome. But remember, unlike mitosis, cells moving through meiosis II are haploid, and the sister chromatids are no longer genetically identical. Now let me show you just a small window into the genetic variety among gametes that results from meiosis. In this slide, we have a homologous pair with a gene for a characteristic, and the alleles for the gene are different. When the alleles segregate during anaphase of meiosis I, the resulting cells are genetically different, as can be seen here. Some of the sperm carry this allele, and some of the sperm carry this allele. Lastly, this slide reminds you that the DNA replicates prior to prophase I of meiosis. You should have a reasonable grasp of these IB syllabus statements. Diploid nuclei have pairs of homologous chromosomes. Haploid nuclei have one chromosome of each pair. Separation of pairs of homologous chromosomes in the first division of meiosis halves the chromosome number. Now, I've come back to this graph because I think it's an important approach toward outlining and understanding meiosis. Hopefully, you now fully understand the nature of gametes as haploid cells with half the number of chromosomes, thus half the amount of DNA per cell. But think carefully about the DNA content after meiosis I. Here, each of the two cells has the same DNA content per cell as the original cell. But here, each cell is haploid with half the number of chromosomes. Now, how is this possible that the cells here have the same DNA content per cell as the cell here? Each cell here has two chromatid chromosomes. So if you think of this cell here as having 46 one-chromatid chromosomes, the cells here each have 23 two-chromatid chromosomes. Thus, the DNA content per cell is the same, but genetically, the cells here are different from each other and very different from the original cell. So here's the relevant IB syllabus statement, which you should be able to address. Outline the stages of meiosis, clearly stating the events of each stage. Mention should be made of homologous pairs of chromosomes, two divisions, diploid and haploid cells, and the outcome of meiosis. In this slide, I compare mitosis and meiosis with respect to chromosome number. Examine the slide carefully. The cell here is diploid, 2N, with 4 chromosomes. Each mitosis and meiosis is preceded by DNA replication. Then, notice the difference in the lineup of chromosomes at metaphase of mitosis with metaphase I of meiosis. In metaphase I of meiosis, homologous pairs are aligned together. Notice that each of these cells is still diploid with 4 chromosomes. But notice that the first division of meiosis results in 2 cells, each with half the number of chromosomes, haploid, 2 chromosomes each. Mitosis results in 2 cells, both diploid, each with 4 chromosomes. Meiosis, because of its 2 divisions, results in 4 cells, all haploid, 2 chromosomes. And remember, these gametes are genetically different from one another. Here is another slide that compares mitosis and meiosis with respect to chromosome number. Study this slide carefully on your own. If you were struggling 5 slides ago to understand the relationship of chromosome number and DNA content through meiosis, here's a slide that can help. Let's follow chromatids, for lack of a better word. On a per-cell basis, there are 4 chromatids here, 8 chromatids here following replication, 4 chromatids here, and 2 chromatids here. Let's follow chromosome number. 4 chromosomes, 4 chromosomes, 2 chromosomes, 2 chromosomes. Study this slide carefully if you're not sure whether or not you have control of meiosis. You've seen this IB syllabus statement before, but I want to introduce you to the random orientation of homologous pairs in metaphase 1. Explain how meiosis promotes genetic variety in gametes through crossing over in prophase 1 and random orientation of homologous pairs in metaphase 1. Keep in mind that I've already introduced aspects of crossing over, such as when it occurs in prophase 1, and that it results in sister chromatids that are no longer genetically identical. Independent assortment, or the random orientation of homologous pairs in metaphase 1, and crossover will be covered in detail in Meiosis Part II. As the homologous pairs arrive to the metaphase plate of the cell, the pairs could arrive in one of two positions: homolog 1 on this side with homolog 2 on this side, or homolog 2 on this side with homolog 1 on this side. And the positioning of one pair is independent of the positioning of the second pair. If you remember, the chromosomes of a homologous pair are different from each other because of the different alleles of a gene. Let's take a closer look at how the random positioning of homologous pairs relative to one another creates genetic variety in gametes. In this diagram, you can see that there are two possible arrangements of the two pairs. We have this arrangement or this arrangement. Notice the alleles and how the positioning of the homologous pairs relative to the other pair would result in genetic variety, even in this simple diagram with only two pairs of chromosomes. The arrangement of pairs in the top diagram results in these two genetic types, while the arrangement of pairs in the bottom diagram results in these two genetic types. In the sexual organs of an individual, where numerous meiotic divisions are producing millions of gametes, all possible genetic types occur. There are two things to note here. I've only taken meiosis through the first division. The second division separates chromatids, so we don't need to look at the second division to see the importance of the random orientation of pairs prior to separation in meiosis 1. Secondly, I want you to imagine 23 pairs rather than just 2 pairs. The number of possible orientations of homologous pairs is 2 to the 23rd power, just under 10 million different possible arrangements. So maybe you can start to appreciate why siblings never look exactly alike. Again, you can follow the alleles noted through the two possible positions of homologous pairs. With only 2 pairs of chromosomes, there are 4 genetically different gametes produced, but imagine 23 pairs. The number of possible arrangements at the metaphase plate is 2 to the 23rd power. Random assortment is the random orientation of homologous pairs in metaphase 1 that promotes genetic variation among gametes. As a summary of the sources of genetic variation, here is a table. Meiosis results in genetic variety through crossing over between homologous chromosomes during prophase 1 and independent assortment of homologous chromosomes during metaphase 1. After meiosis, random fertilization of an egg cell by a sperm cell, aka random mating, also results in additional variation. Can you state this? State that the fusion of gametes from different parents, random mating, promotes genetic variation. As you look at homologous pairs here, remember that they carry genes for the same characteristics, but the alleles for the gene may be different. Remember that one of each homologous pair has arrived in the sperm and the other of the pair in the egg. New combinations of alleles are brought together when people meet and mate. The fusion of gametes from different parents promotes genetic variation. And that brings us to the end of Meiosis Part I.